Our webinar will start at 10 a.m. sharp. Please stay tuned with us. Thank you. Testing. All right. A very good morning to everyone. Welcome to the webinar titled Cyanobacteria, Natural Products and Their Biosynthetic Genes. The webinar. My name is T. Kim Boon. I am the moderator for this webinar. This webinar is co-organized by Postgraduate Society, Faculty of Pharmacy, Universiti Malaya, and Center for Natural Products Research and Drugs Discoveries, CENAR, Universiti Malaya. The webinar is conducted using Zoom and also Faculty of Pharmacy Facebook Live. You can share the Facebook Live to your friends who miss out the registrations. The link is in the chat box. Okay, let me explain the tentative flow for today. The webinar is started with a lecture. There is a short break after half an hour for Q&A session. You may type your question in the Q&A box or wait until Q&A session starts to raise your hand to ask the question directly to the speaker. For Facebook Live audience, you can post your questions at the Facebook page. Our committee member will send the questions to the speaker here. After this, we will have a video presentations for 5 minutes. Then we will continue the lecture. We will post the feedback form in the middle and at the end at the Zoom chat box as well as the Facebook Live page. Please fill in the feedback form for our improvement. The committee will email the e-certificate to you later. The feedback form will be closed at 12 p.m. today. So no certificate will be issued if there's any entry or the entry is not captured in the feedback form. Okay. Today, we are glad to have our distinguished speaker, Dr. Phan, to share his valuable knowledge on cyanobacterial natural products with us. Let me introduce Dr. Phan. Dr. Phan was born in Malaysia, and he is a big fan of NMR spectroscopy. At 2018, he obtained PhD from Universiti Malaysia Sabah for investigating marine soft coral-derived natural products. During 2018 to 2019, as visiting fellow at University of Western Australia, where he first exposed to natural products biosynthetic pathway. He then been awarded a JSPS Postdoctoral Fellowship 2019 to 2021 at Hokkaido University for investigating cyanobacteria peptides. Also, he just started his career as a research fellow at National University of Singapore this week. Without further ado, let me invite Dr. Chin. Dr. Chin, the floor is yours.
Yeah, thank you very much. I will share my slide.
Yes.
I can share my slide. Can you hear me?
Yes, I can hear you. Yeah, our technical team is preparing now.
I can share my slide right now.
Okay, we can see the slide, but not in presentation mode yet. Yes, it's in presentation.
It's good
Everything
now?
is fine. Yes.
All right. Please wait. Yeah, thank you very much for inviting me to share my work at here. So today I'm going to talk about cyanobacterial natural products and their biosynthetic genes. So as Kim Boon have given a generous introduction about me, so I would like to introduce myself again. So let me make a laser point. So I get my PhD under Professor Charles from UMS 3 years ago, working on isolation of natural products from marine organisms. So one of our goals is to discover new natural products. And that time, our chances to encounter new natural product is about 30%, which is quite high because our location is blessed with high biodiversity of marine organisms, which means there's a lot of chemical communication, and we really love that. So what got me into the natural product? So this is Professor Charles. He is a top research scientist in Malaysia last year. I was inspired by his lecture about natural products, and then I become interested in MR spectroscopy. So actually, Professor Charles is right now with us. He's one of the, he's now with us as an audience in this talk. So because of the MR allow me to see the world in a chemical structure way, and that's how I start my PhD study under Professor Charles. Like we can see plants, chlorophyll, and lemon, vitamin C, and the gene, the day agent. Yep. So after that, I went to the University of Western Australia, one of the top 100 universities in the world, as a visiting fellow. I started to wonder how nature could produce such diverse chemical structure and how their stereochemistry is controlled by nature. For example, in here, the stereochemistry is S configuration. So why? Who decides this? So that's the question we began to ask by ourselves when I was in Australia this year. Then I moved to Japan, Hokkaido University, after being awarded with JSPI postdoc fellowship, where I continue to interrogate nature, the origin of life, which is genome, to connect chemistry to genes. And now I'm a research fellow at NUS, one of the top 20 universities in the world, to do some cool stuff. So that's about me. So let me give you a bit about natural products. So you see here, this is the ocean, and it's covered by a green stuff. And this green stuff actually a microorganism is called Cyanobacteria. So this phenomena of overgrowth of Cyanobacteria is called red tide or agar bloom. So when they overgrow, this Cyanobacteria produce toxin. This toxin is very poison to both aquatic life and human being. So this toxin is not from primary metabolism. So talk about primary metabolism. Let's go back to biochemistry. So I'm sure that everyone knows this cycle. So all life forms do these processes in a very similar way, and them are fundamental to life, as we have learned this during the high school. Yes, this is a Krebs cycle. It helps to release energy from complex molecules such as carbohydrate. This is a primary metabolism. In the process of doing this, it creates a whole series of intermediates. So the toxin I'm talking about is not from primary metabolism. It's produced by secondary metabolism. And secondary metabolism is in fact this field of natural product chemistry. Living organisms can make molecules outside of primary metabolism. It's called secondary metabolites or natural products. These molecules are basically built by stealing this type of intermediate from the primary metabolism and incorporating them into a much complex structure. So so this idea about primary metabolism and go to the biochemistry and get cycle is actually from Professor Paul Jensen from UCSD, University of California, San Diego. So to continue, so these are the cyanotoxin are produced by the secondary metabolites from the cyanobacteria. So a bunch of them you can observe during the event of red tide. So these are the toxins that killing a lot of fish and is poison to human if you drink the water. I mean, So people have been asking, what are these toxins for? So a lot of investigation, application, finally people believe that this toxin play an important ecological role in chemical defense. So let me give you another example of natural product that play an ecological role. So this natural product is called anochelin, and it's seedable. It's used to ion uptake for the cyanobacteria. So what happened here is that this is cyanobacteria cells, and first it releases the natural product, this chemical structure, this molecule. This seedable is represented by blue sugar into the environment, which is ocean. So in the ocean, there are free iron, which is represented in red sugar. So the seedable will have a strong affinity bind to the red iron and can be uptake by the cyanobacterial membrane into the cells, and where it can release the iron for the cellular process in the cyanobacterial reaction. So metals such as iron is very important cofactor for many reactions. So let me show you one example. So inside the cyanobacterial cells, chemical reaction is catalyzed by enzyme, or we call protein, where the cofactors such as magnesium ion, in this case, to facilitate diphosphate ionization, which create a positive, or a cation adhere to initiate electrophilic reaction. And as a result, the branny group, this 5-carbon branny group, add to this molecule for the cyanobacteria function. So this natural product with the function of ion uptake by cyanobacteria has the application in antimicrobial biomaterial to make hard stand used in many patients with problem of too much cholesterol. So the natural product that is original function is for the ion uptake ecological role for the cyanobacteria to survive in the harsh environment. It has been discovered have the benefit for human to making a hard stand. So as you can see, the example, the seedable can be used in medical tools for the human benefit. Other cyanobacteria natural product also being used in many drug discovery programs, such as this atoboinin one, which is published in this year, PNS, is a promising anti-cancer candidate. And there's a tulastantin 10, is a promising anti-cancer agent, and now have been developed into clinical trial. Undergo in the clinical trial, and most likely will develop into the drug. And it is clear that this example that I show you and the previous example that I show you, that these natural products are not made to help humans to fight against disease. This type of natural products actually play an important ecological role, such as chemical defense, or competition and chemical communication. It is simply that we have discovered our own application for these natural products. So as I have shown you, natural product has been a great benefit in medicine discovery for drug discovery. So we also can say that natural products, I mean, without natural products, many people will be starved, hungry, as it can be used in agrochemical field as a crop protection to protect the crops from the insect or pesticide or other thing. Yeah. So besides medicine and food, natural product can be used in materials to satisfy human desires, such as perfumes or painting. So there's a lot of benefit for using natural product in our life. So as we talking about cyanobacterial natural product, I really have to introduce two guys who are the pioneer study of cyanobacterial natural product in the early years. So this is the late Professor Richard Moore. He's an emeritus professor in University of Hawaii in United States. He started cyanobacterial natural product in the very early, about mid-18th century. And also at the same time, not at the same time, a little bit later, Professor Bill Garwick from the University, he's the current professor at the University of California, San Diego. He's still very active in cyanobacterial natural product. And these are the two pioneer study for natural product. In fact, actually, I got the attendance list, participant list for this talk, and I saw one of the guy. His name is Tiago Leon. If I'm not sure I pronounced his name correctly, he has spent some time in Bill Garwick laboratory, and he's the participant of this talk. Them really do a really cool stuff and making a lot of great paper. And Tiago Leon is, I think, I briefly checked, he's a postdoc right now. He's made good paper like PNNS and natural metabolism. Yeah. So I will, yes. So to continue my talk. So now we go into the cyanobacteria natural products, and I have to describe.
Yes, Dr. Chin. Yeah.
Kim Boon, do you want to stop to?
Yeah, we will stop to rest for a while for the video sessions. Yeah. Thank you. Can you stop share the screen? Yeah, okay, thank you. So thank you, Dr. Phan, for the first sharing on the introductions of the cyanobacteria to different fields and some basics and knowledge about cyanobacteria. So now we will stop for a while to check any question in the Q&A box. So from here, we don't have any question at the moment. And we will proceed to short video about Faculty of Pharmacy and also CENAR services. Yeah.
Welcome to Faculty of Pharmacy, University Malaya. are committed to the provision and delivery of the best services to our stakeholders.
The next video will be the CENAR University Malaya. Just hold on for a moment.
Hi everyone. Our center is named the Center for Natural Products Research and Drug Discovery. It has been established since 2003 by several of the leading professors who have worked in natural product research. This has become a platform for all natural product research at the Universiti Malaya. So we offer several services. It ranges from extraction, isolation, purification, or plant extraction. We do offer compound identification from structural elucidation. We also do biological assays, for example, antioxidants, anti-cancers, antimicrobes and many more, especially in vitro studies. We have high-end instruments analysis that are NMR, GCMS, and LCMS. We do have these instruments for quantification and method development such as HPLC and LCMS. We do docking for drug design and development. We welcome and accept research attachment and student internship from local and international. So, where are we located? We are located at Level 3, Research Management and Innovation Complex, Universiti Malaya. For more information and inquiries about our services, you may contact us through phone or email at cenar@edu.my. Don't forget to like and follow our Facebook page. See you soon.
Thank you for watching the video. So now we will continue the lecture together with Dr. Phan.
All right, I will continue to share my screen. It's good? Everything good?
Yes.
All right. Yes.
It's in the viewer mode. Can you put on the presentation mode?
Oh, okay. Please wait. How do we do that?
Yes, now it's better. Yeah. Now it's in, yes, it's in presentation mode. Yes, good.
So I have given you guys the background of cyanobacteria, natural product, or also the primary metabolite and also the secondary metabolites. So now you know how the secondary metabolite came from and what's the purpose of secondary metabolite to the organism, but also can be used for the human well-being. And these are the two pioneer study for cyanobacteria natural product. So now I want to go into the genomic stuff for the cyanobacteria natural product. So there are a lot of genomic stuff. It's a big field. So but right now I just want to talk about rates. So rates is what we call ribosomally synthesized and post-translationally modified peptides. So it's a large group of natural products where they display a distinct chemical modification on the precursor peptide. I will talk about this later. So RIPS is kind of like a big group of natural product. It's a it's a big group, but there are a lot of big group in genomic natural products. So in this big group, we are going to focus on small family, what we call Cyanobacteria. So Cyanobacteria is a family of lips natural product produced by symbiotic and free-living Cyanobacteria. So the background of Cyanobacteria, how it's produced. So from the genome, once we sequence the Cyanobacteria genome, the DNA, what we do is we do carry out the annotation on the genome sequence. So after that, we will get a biosynthetic gene cluster. So after annotation it means it's analyze the DNA sequence and interpret and try to interpret that the origin of DNA sequence into a protein or genes. For example, here this open reading frame have 1, 2, 3, 4 have seven genes. So like this, the first gene, like purple color, is part A. That's what people give their name, and they are part D, E, F, and G. So let's focus on part E, this gene, red color. So this gene is a precursor peptide. So this gene have the origin of DNA sequence at here, SO at here, ATG, AAC, AAA. Yeah. So as you guys have learned during the high school, the what do we call central dogma, you know, from DNA to mRNA and then to protein. That's what we learned from the high school, right? So the PEG, the gene, is a precursor peptide. It has the DNA sequence of this here, and it can be transcribed to mRNA, and then it can be translated to a polypeptide, a protein polypeptide, as like this. You can see the structure of polypeptide of this. So this PEG basically encoding polypeptide of this. So what happened next for the cyanobacteria is, so this polypeptide is then modified by the PEG. PEG is a heterocyclase. So you can see from here, you see the what do you call it? This is a 12-9 amino acid. It can be modified into this 5-member link, heterocycle link. So this PEG will modify all along the sequence of the amino acids, the polypeptide. So as long as it detects the 12-9 and also the serine, it will convert into a heterocycle. So this is the function of this gene, PEG. So the other gene, for example, PEG, is an N-terminal protease. So what happened here is the PEG will cut off the N-terminal, and here you can see this initial polypeptide has been cut off, and here has been cut off. So now you got this, and then it will continue to process by another gene like PEG. So PEG is a C-terminal protease and macrocyclase. What he do is he cut off this AYD, AYDGE, and I make them into a cyclic peptide like this. So this is the final product of this biosynthetic gene cluster, of this genome, of this DNA sequence in the life of Cyanobacteria. That's how the secret of life, the origin of life, how the molecules are contained in the DNA sequence, and the DNA sequence is making this type of molecule like patolamide A and patolamide C, which holding a great ecological role for the survival of Cyanobacteria. And this has been published in a very good paper, PNAS, in 2005. So the last step of the Cyanobacteria, this family natural product, is the PEG-F this one. So PEF is a penny transferase. This is the crystal structure of this protein, PEF. So what happens is PEF will bind to this structure and adding a penny group here, 5 carbon penny group, like this 5 carbon chain. So to match, to modify the final product into this product, so what we call penny algalamide. So there's a reason why the cyanobacteria enroll for the existence of this gene to adding this, the red color you can see here, a 5 carbon chain into the natural product, because it can enhance the activity, the ecological load for the survival of cyanobacteria. Okay. So the biosynthesis of the cyanobacteria, starting with the E family protein, which is this one, is a precursor peptide, just as I described to you, from the DNA sequence to mRNA and polypeptide. And this polypeptide is continued to modify by D family protein here for the heterocyclase, where this protein encounters 309, it will make its heterocyclase. It's N-terminal protease. It cut off the N-terminal region, the initial polypeptide region, and also the G family protein will cut off the N-terminal, the C-terminal, the end of the polypeptide region, and make a cyclic peptide. So finally, the last process is the F family protein, splenic transferase. So cyanobacteria is a peptide with a distinct function with a penylation on the natural product. So whenever you see a cyclic peptide produced by cyanobacteria with a penylation, that is identified as a cyanobacteria. So this is how the origin biosynthesis pathway of the cyanobacteria, and published by PNAS 2016. So now I'm going to share what I do in Hokkaido University, Japan. So you can see this. This is our department. It's very small, only 2 floors. And this is during the winter. There's a lot of snow and it's really freezing out here. And see this picture. It's how we grow, how we culture the cyanobacteria in our laboratory. So I want to introduce you the Cyanobacteria microcystis aeruginosa, NIS 88. So NIS is stand for, I don't know what it stands for, but NIS is a microbial collection in Tokyo, in Japan. So people around the world can actually buy the Cyanobacteria strain from the NIS collection. The price is about 400 ringgit Malaysia for one specimen, for one sample, one strain, one Cyanobacteria strain. So this is Microcystis aeruginosa, and this is how it looks like under the microscope. So what this Cyanobacteria strain has been well studied seen in the very early 19th century, and you can see that there's a lot of papers have been published about this strain because people keep finding a new natural product from this strain, like Microcystis LR, mycopectin, and mycopectin again, and also Kawaguchi pectin. So this strain is very famous around the world, as Microcystis aeruginosa produce a lot of natural product and produce a lot of diverse natural product. So what we found here is we found there are 3 peaks as shown in this LCMS profile, a peak 1, peak 2, peak 3, and we found that peak 1 and peak 2 has a difference of 68 Delton, and 2 and 3 also have the same 68 atomic mass unit different. So I know that 68 can be represent this type of 5 carbon chains, what we call planning group. So we quickly checked the literature, and we found there are 2 compounds just like I showed you. Microcystic 88 have been reported to produce Kawaguchi-Pectin A and Kawaguchi-Pectin B. And you can see A and B, the difference is additional 5 carbon chain here and here. So we suspect that, not we suspect, we believe that we have found another type of cyanobacteria with a ventilation here, 68. So like big 1 is a bis benylated compound, and big 2 is a mono benylated, only 1 benylation, and big 1 is 2 benylation, and big 3 is no benylation like this one. So that is what we believe. So we quickly proceed with isolation and purification, and we found that the structure of the compound we have discovered is the arginine A, B, and C, and these are the new natural products we have discovered from this well-studied strand. So we are really surprised that how come nobody have discovered this when this is a well-studied strand. And we quickly do the MR structure. So this is the MR data, and also we identify the absolute configuration using the MAFRE analysis to determine the amino acids configuration in this structure. You can see here. And also we found that there's a cis poli and trans poli in this structure. So we done all that, and we really accept that as the reason why a well-studied strand have this type of unreported compounds. Is not only is a new compound, there's a no well moiety at here. So we really excited about this. So as I have learned the genomic study during the Australia, which is a turning point for me and get into the genomic stuff. So I was actually under Professor Tatsufumi Ogino in environmental science department, where we do isolation and chemistry stuff for my JSPS fellowship. So I quickly proposed that I want to do a genomic stuff, not only chemistry, but I want to put my 50% of my effort into genomic stuff. So we quickly found our collaborator, Professor Toshiyuki Wakimoto. So he's a famous guy in biosynthesis. So we go to him in the pharmacy department. He's doing the genomic stuff, and we quickly collaborate with him. So I spend time in this laboratory, and also this laboratory, I go there and there between always, and this is what we produce. So this is the paper. It's me and also Wakimoto, Professor Wakimoto and Professor Ogino. So this work has published in one of the most prestigious journal in all chemistry, Journal of American Chemical Society, with an impact factor of 15.4 in this year. So what we do after I went to the Faculty of Pharmacy and doing the genomic stuff is that first we check the sequence as I show you. This strand has been well studied, and there is a draft genome that has been published in the NCBI database. It's a public available genome data. So we check, this is the previous study published in Angers. This is #1 chemistry journal in Germany. So this is the actual NCBI database number. So we found, and this is the vanillin transferase, but we could not find the structure that encoding the precursor peptide that encoding this, the core peptide of this structure. And we can see here, poli is represented by P, poli P, and arginine is represented by R. So we quickly find this code in the genome data, and we cannot find the precursor peptide of this So what we do is we sequence again this strand, and we believe that the data that I published in this is a draft genome might have missing or misassembly when they publish this data. So we do again, sequence our data again, and we found the missing part as you saw. This is not a draft genome. This is a complete genome data of this strand. So we found the missing part of this genome, this DNA region, and we found the precursor peptide. So now we know the biosynthetic gene cluster that produce this chemistry. Basically, what we have achieved is that we have success to connect the chemistry to genes. So here I want to propose the biosynthetic pathway of the compound that we have discovered. So as I mentioned, this strand has been well studied. One of the papers that just now I published here, Ange 2016, with the draft genome. This guy have reported the Kawaguchi peptide, the biosynthetic pathway, in the one of the paper in Ange, as it is #1 paper in chemistry. #1 paper in Germany. So this is the gene cluster. He has been characterized. So our gene cluster is here. It's very far away, as represented by this symbol. So as I mentioned, this the E family protein is a precursor peptide, which encoding the polypeptide of this. And here is a core peptide. So what happened here is, so AGCE hijack KGB-A. And just now I mentioned, A family protein is the N-terminal protease. So he cut off this initial peptide, what we call leader peptide. So we cut off this here by A family protein, which is KGB-A. Of course, this gene cluster do not have AGCA, do not have A protein. It only have E and F protein. So this is unusual feature that it hijack a gene cluster from another gene cluster far away, like here. So hijack that one and cut off here, and it continue hijack another gene, G family protein, cut off the C terminal, this region, and make a cyclic product. That's the process of this. So after that, after the core peptide is processed by KGB-A and KGB-G, you get a cyclic product, which is the compound trait azithromycin C. And it can be further processed by AGCF. AGCF have been annotated as a benzyl transferase. And what the function do is benylation, adding this 5-carbon red color chains into the this moiety, guanidine moiety. So as you can see, azithromycin C have an antibacterial MIC more than 100 micromolar. But when adding of this 5-carbon chains red color, the antibacterial activity has increased a bit. So, but if further process, this AGCS is very unique. It doing 2 rounds of benylation, not 1 round. Usually 1 round, but this unusual feature of this protein, AGCF, doing 2 rounds. So making a base benylation on the guanidine moiety, making this product azithromycin A, which is compound 1. So it have a really good antibacterial activity. So you can see here, as this red color thing attached to the natural product and to the molecule, the antibiotic activity increases. So that's what this work has published in this JAS. So to prove that our proposal of the biosynthetic pathway, we do a heterologous expression. So basically we express AGCE or AGCE plus KGB-A or plus KGBG, but there's nothing produced. But once we co-express a gene of AGCE and KGBA and G, we found a single peak. And this peak is belong to compound 3, as you might see. And there's a co-injection and everything is consistent, and we have proposed that the biosynthetic pathway of the azithromycin compound is from AGCE, and it can be processed by hijacking faraway gene such as KGBA and KGBG to make a mature product. So to further verify our proposed pathway, we do an in vitro assay, in vitro activity of KGBG. So what we do is we express this gene KGBG and purify this protein KGBG. And so what we do is just now the heterocross expression is everything inside the E. coli cells. We take out the gene and put inside the E. coli and let the E. coli to produce this compound, this molecule, where this molecule is originally produced by Cyanobacteria, Mycosis aureus. So we put inside a different host and let it produce. It still produces the same thing, and this has proved our hypothesis. But really have to verify this, we do additional experiment like in vitro assay. So what we mean is that we are not using E. coli anymore. We take out everything and put inside an artificial tube and see the conversion of compound 3. So it's no longer inside living cells. It's just a free base experiment. So we purify the protein and put inside a tube, and we mix with this compound, this structure, this molecule. We call the thing, and you can see this one is really like this. So you remember just now the red color stuff here. So we make this, we synthesize this, and this is actually something like this, same thing. So we put this and mix this together, and we found that when we mix this together after 16 hours, it produces this peak, which is compound 3 standard. So without mixing this compound 13 here, and with just only the enzyme KGBG, the protein, there's nothing. So this has this conclusion that support our proposed pathway, that this critical peptide actually can hijack in an unusual way from the faraway biosynthetic gene cluster to process the precursor peptide into a mature molecule such as this one. So this is unusual phenomenon, and this only have one example from the previous study, which is published by last year. So usually, when we talk about biosynthetic gene cluster, usually it's a co-linearity rule. What it means is that everything is clustered together. So that's why we call biosynthetic gene cluster together, near to each other. So there's one example of lakes, this seedling air, where the precursor peptide also been processed by hijacking faraway proteins like this one. So that's one example. So it's like we are second example. Yeah. So as I showed, we have already finished the AGCE precursor peptide. We want to know about AGCF. This protein is a pentytransferase. We want to prove our hypothesis is correct, that this AGCF can do the penetration on here, and second round penetration at here. So what can we do is we do the same thing, the in vitro assay. So cell-free, outside of cell. So we purify the protein AGCF and mix with the compound 3 as a precursor, no, as a substrate. So, and we do a time-based assay, as you can see at 0 minutes, with the protein and the substrate compound tree. There's no change almost. And after 1 minute, you can see a small compound 2 is increasing, and 5 minutes is more. And as the compound 2 is increasing, it becomes a substrate for compound 1. And you see at 2 hours, there's a complete conversion to produce a base ventilation. And if we're using a boy protein, I mean the non-function protein, there's no conversion. So this really has shown that we have succeeded to show people that we have found a protein HCF can do an unusual 2 rounds of ventilation at this moiety arginine. So a little bit about the background of characterized cyanobacterial transferase. So far that what we have seen, people have done is you see this benny group on the thyroxine or all the threonine or serine or all the tryptophan residue. That's all. So what we really found that we found a protein of benny transferase that can catalyze a benny group on the arginine. So this is really a new feature of the benny transferase. This is really a good discovery because as I've shown here, until now, what people have discovered is thyroxine and tryptophan and threonine or serine. So this is a new feature. We have filled in one gap closer of the benny transferase. So this is a phylogenetic analysis of the primary sequence of the benny transferase. You can see here we form a crack of here. It was separated from all the other benny transferase. And you may be wondering why this red dot at here. And this red dot is very closely to this HGCF. It's a same crack. We can say a same thing, same protein. So this red dot represents our group is now doing this. So it's a timeline of characterized cyanobacterial benny transferase. So back into the 2011, this is the first cyanobacterial benny transferase have been discovered and characterized until here. So this is our discovery 10 years later, which is now. So during this time frame, within this 10 years, the benny transferase have been increased the popularity. People have been doing this. It's really interesting. And you can see that this benny transferase can accept the benny donor of the MLPP, carbon 5. Just now I show you the chance. Like this one have 5 carbon, like this 5 carbon. And there's a GPP. GPP have a 10 carbon, like here. So during this time frame, the only reported the benny transferase only have 1 can accept the GPP donor. The last is only 5 carbon donor. So we're asking ourselves, is this GPP is possible for our benny transferase? So we have shown in the vitro assay, we have shown with enough evidence and data that it can accept the MLPP in 2 rounds of ventilation. So we do additional experiment in vitro activity again for the protein with 2 different benny donors. One is GBB, carbon 10, and another is FPP, carbon 15. So to our surprise, as I show you here, the benny transferase is really specific. Then only can accept 5 carbon benny group. Only 5 carbon, 5 carbon, 5 carbon, 5 carbon, or 10 carbon. Usually it's like this. It is not possible to accept both 5 carbon and 10 carbon. It cannot be done there until now. But in our unique feature of our binary transferase that we have discovered, it managed to make it was not efficient, but it's managed to make a small mono GPP product. As you see the structure here, it can accept this vinegar, and because the amount is very small, we cannot do the MMR structure. What we do is the MMS to support our conclusion. So after that, we want to characterize the chemoselectivity of this protein. So what we do is we synthesize this substrate and this substrate. So we can see the red colors. This is the catalytic, the acetylene residue of this the substrate. So the original is arginine. So we prepare tryptophan, serine, lysine, threonine, and thyroxine. And there's a linear structure also. So our justification to design this synthetic analog is based on this. So far, the characterized binary transferase has been a thyroxine, a threonine, a tryptophan, and also a serine. So we want to see, is it really our binary transferase different from all the other characterized binary transferase? Is it our binary transferase can only be accept this type of structure with arginine? Can it be accept thyroxine or threonine or lysine? That's what we interested to find out. So in the conclusion, there's only one changer, which is here. The rest is not accepted by this protein, AGCF. So the conclusion is we have determined the chemoselectivity of this protein, which is arginine. So, and it was not able to accept any linear product, even a very similar structure like compound 11 and 3. So the conclusion is what we have found, the behavior of this protein, AGCS, is there must be a cyclic product. Second, this must be an arginine unit at here. That's the so far we have this characterized of this protein. So why we do this? I mean, why if you look at this time frame from 2011 until now, 2021, 10 years, people have been study about this cyanobacterial binary transferase protein. Why we do all this stuff? So here I'm going to show you peptide is an important candidate for drug discovery. So in this figure, published in this paper, in the last decades, peptide have became a major market in pharmaceutical industry. And because peptide therapeutic have a size, selectivity, and low toxicity. However, peptide has a weakness because it's often susceptible to proteolytic degradation or failure to cross cellular membrane. But this weakness can be overcome by one way to improve this weakness is by lipidation or pennylation. Just now I present to you the protein that can do pennylation. One way to overcome the weakness. So the current method for synthetic peptide alkylation is limited. Also, the discovered enzymatic alkylation also limited. So that's why people are looking for binary transferase. So these are the example of lipidation and pennylation that have a really good benefit in terms of drug discovery. So lipidation as a chemical modification could boost the peptide therapeutic efficacy. So this is the US FAD approved anti-diabetic drug, semaglutide. So this drug is possible because of this lipidation. You can see a similar thing here, looks like a pennylation with 5 carbon, 10 carbon. This is a lot of carbon, maybe 20 carbon chance. Yeah. So granulation is almost same to lipidation. Granulation as a chemical modification tools could enhance peptide biological activity and like this peptide, and has been patented for cytotoxic agent and currently is undergo for drug development. So this is a good example of why people are doing this. So in our paper that published in JAST, what we found is number of granule increase and the antibiotic activity is increased as shown here. So this result is really consistent to what have been done by other people. So finally, I want to talk about the conclusion. So what we have found right now, we have introduced a new tool for peptide modification. And I have shown you this type of modification could really develop into antibiotic drug. So in future, we want to do protein engineering to create a useful biocatalyst. So biocatalyst is this thing, this protein, this kind of like a catalyst from here to here. So we want to make a more useful biocatalyst. Right now, the protein is not perfect. Of course, it's perfect for the cyanobacteria itself, but it's not perfect for human application. So we want to make a biocatalyst. What we imagine is that there's a chemoenzymatic synthesis. What we do is people can make a lot of, synthesize a lot of cyclopeptide like this, a different color cyclopeptide, put inside this jar, fill with the protein of AGCF, let the reaction, let the protein do the job. After a few hours, it can generate a bunch of this of analogs with penetration, and a bunch of this analog, like hundreds or thousands of them, can be done in few hours, and we can screen for antibiotic. This way, it's used as it's like a platform for drug discovery, and some of them are now doing in pharmaceutical industry. So what we propose right now here is what we discover, the size we are working on is a basic size, and there's a trend, basic size go to the applied size. So what we imagine that our protein can be do this way, and it can be used in the pharmaceutical industry in this platform to discover new antibiotic drug, and that is applied size. So we really excited. We're really excited about this prospect, and so keep your eyes out. If it does work, it will be a pretty big deal, because everybody grant proposal that working on this field will be viewed more positively when there are example of drug developed through this type of size. So we are really excited about this, because we can foresee the future benefit of this protein can bring to the human being. We have patented this with Hokkaido University. So we patent this. So when we talk about a pharmaceutical, company or pharmaceutical industry, people will only be interested if there's a patent, because it prevents from other people doing that. So we patent this, and we'll collaborate with industry to make this possible. So we are really excited about that. So I finally a few more slides. I want to talk about the history, the timeline of characterized by cyanobacterial transferase. So I really want to introduce this guy. He's a Professor Eric Smith from University of Utah, United States, America. So he's like my hero. So you can see when I'm doing this work, I always checking his paper from times to times, because he's the pioneer. He's like a father of cyanobacterial transferase. He have done a lot of work on this. You can see he started this back to the 2011. he started this in the first cyanobacterium transferase. He characterized its LYNF, LYNF, this protein. And then he continued to do the work. You can see from this timeline, maybe 70% is belong from his laboratory. So even the group that I will be joined in NUS, also he is one of them at here. So he's really inspired me a lot from his paper. That's how we design the experiment and what he do. We keep looking at his paper here and make our judgment and our next step and hypothesis. Yeah. So I really want to thank him. So finally, I want to dance. I mean, this acknowledgement, so I should thank somebody. So we make, because our collaboration in Hokkaido, we managed to make this paper. So I want to thank my host, JSPA host, Professor Tatsufumi Ogino, and Professor Toshiyuki Wakimoto here. And actually this work is shared with Masuda's sensei, Dr. Masuda. So he is Masuda. And although another two students, Nandani and also Fujita. So in Hokkaido University, if you see here, we have a beer and our dress like this, and there's a lot of sweating. That is because every week we have a jogging for about 10 km. So we always do this and beer, and during that time we talk about science and what is the future like this. And yeah, this really a good time. So I also want to dance about Hokkaido University supporting our work and advertise our work in a press release. And also I want to dance about JSPS postdoc fellowship for funding me and in terms of salary, and also Takenhi in terms of research grant about this amount. So just like many other grant application, there's a outreaching component. So what it means outreaching component is that they want us to talk to people like you and tell you what we are doing. So that's the grant. Most grant have this component. So that's why I'm here today. Yeah, I'm finished. I'm happy to take any question if there's any. Yep.
Yeah, thank you, Dr. Phan, for your impressive and remarkable work. We have received many positive feedback from the audience. Yeah. So now we have 7 questions from the Q&A box. Shall I read for you?
Oh, yeah, sure.
All right. So let's go to the first questions. Morning. Is there any research relating to cyanobacteria and medical intervention of autoimmune diseases?
So when we talk about autoimmune diseases, I believe that is one of them is like inflammation. So I'm sure some of these cyanobacterial natural products can have a anti-inflammatory activity, and some, not technically in the drug market, there's an anti-inflammatory drug, but some of them show some of this activity, and I don't have a lot of knowledge about this, but I know someone is doing that in the NUS, National University of Singapore, doing this cyanobacterial natural product and also the disease and also the medical intervention. Yeah.
All right.
I hope I can answer this question in more.
Yeah, this is a very general question. I think maybe in the future there will be many. Yeah.
Yeah. If you have more specific question, you can drop me an email. You can easily contact me by just Google my name. Yeah.
All right. So we will go to the next question. Is there a standard practice in the field to understand the molecular mechanism of the anti-disease property of natural products? From what I have seen, it seems that a lot of studies on the anti-disease properties of natural products tend to stop at in vitro phenotypic assays, and rarely do they go thereafter.
That's a good question. This is more like a drug discovery question. So what basically we do is many people are doing is first of course we do the in vitro assay, in vitro phenotype assay, and found there's a good activity. Then only we do in vivo or go into the clinical trial. Of course, a lot of natural product cannot go further because the activity is not strong, or many other reasons, such as dissolvability of this compound cannot cross the brain barrier membrane, and also there's a toxicity of this compound, so that's why it cannot further study, investigate, and go further beyond for the drug discovery process. So what really people do right now is there are some group in the United States of America, them study the known natural product. For example, the compound that have not as good activity, have a weak activity in terms of in vitro assay. What them do is modify the natural product in a chemical synthesis way or enzymatic way. Like just now, like this slide I show you, the chemoenzymatic assay. So they modify the structure and then do again the assay, and if they found there's a hit compound, they will be further develop and go into clinical trial. For as far as I know, them are doing, and there's no example I can show you right now.
Yeah, I think you have shared a lot of examples for the audience about the idea how to go further. Yeah. Okay, so I will go to the next question is from Dr. Phan. I am curious to know how the cyanobacteria decide to induce KGPG to synthesize agile cyanomides. It is due to physiological stress? If so, how do one standardize the biosynthesis, say in a bioreactor, or the cyanobacteria to mass produce the agile cyanamide?
Yeah, that's really a good question. I never thought about that. So give me some time.
Yeah, this will be more to mass production questions in the later part.
So of course we have been questioned a little bit about this. After we jogging, sometimes we discuss about this. Why? There's a lot of things. I mean, of course, we publish a really good paper. It accepted in a really good paper, but that is not everything. What we have report is basically one gene, which is AGCF, the benzyl transferase. That's the main thing we discovered in that paper that published in JAST. There's a lot of stuff like what the Dr. Phan, I hope I didn't mention incorrectly his name. This is one of the questions we didn't solve. So yes, we don't know why this cyanobacteria behave this way. Like I mentioned in the biosynthesis, the gene cluster, why the name biosynthetic gene cluster have a cluster behind, and that is because all the genes are clustered together to make a molecule. That's why it's called cluster at the behind of this term. So it's really follow the co-linearity rule. It's a linear, like 1 to 2, 2 to 3. So stick together. We don't know why it's hijack KGBG, for example, far away from a different gene cluster to synthesize this arginine. We don't know, and we don't think it's under stress. I think it's perfectly fine. The cyanobacteria is good. We we provide a lot of nutrient, and he is safe. He is have a lot of nutrient. I think there's no stress. I don't know why he do that. But if you want to do a if you want to do a mass production of argiceramides, yeah, we can do that by cloning the precursor peptide AGCE together with the KGB-A and KGB-G in the E. coli. We can culture mass culture and get a lot of argiceramide. Yep, you can do that, or collaborate with our group in Hokkaido University, Japan. Yeah.
Yeah, thank you, Dr. Phan, for your answer to this. Yeah, we need more research on this. Move on to the next questions. What is your hypothesis of why the collinearity rule is violated in your studies, and the other one you cited?
Yeah, this is another good question. It's almost like just now I answered the question. So yeah, collinearity rule is a linear, it's a stick together, it's cluster, everything is just side by side. But we don't know, as I say, we don't know why, it doesn't follow the collinearity rule that have been established by the biosynthetic expert. We don't know why he do that. I mean, this cyanobacteria. But yes, we have found one example of that, and to support our proposal.
Okay.
I don't know why he do that. So you know that many organisms, including human beings, undergo a million years of evolution to shape what we look like today. So the bacteria or cyanobacteria or fungi do the same way to survive in the harsh environment, competitive environment. There's evolution in the genetic information, the DNA, to make molecule. For example, the fungi to make molecule to protect themselves from bacteria and to do infection on the host organism in order to survive. So this evolution, we cannot ignore this evolution. This evolution have there's a purpose for this evolution for the organism to survive in the harsh environment. So this is like a fitness of the organism. So there must be a reason why the argiceramide, the precursor peptide AGCE, is obstruct the collinearity rule, doesn't follow this rule, and hijack the KGB-A and KGB-G from far away gene cluster. We just don't know why, but there must be a reason, ecological reason, evolutionary reason for this. But for now, I don't know why. But if you have some idea, we are welcome you to cooperate with us in Hokkaido University, Japan.
Yeah, thanks, Dr. Phan, for sharing this. Yeah, we are sure you can find the reason behind later on. Okay, next from Kathleen. Any latest research of chlorQ and NPHP in pharmaceutical research?
Oh, you asking a really specific question. I don't even know what is chlorQ and NPHP, but I assume it's a protein because from this the name of the code, I assume it's a protein, it's a gene. Let assume this benzyl transferase protein. So so I believe I never see chlorQ and NPHP. I assume this is a benzyl transferase protein, but it's not from cyanobacteria. I think it's from other organisms like fungi or bacteria. So of course, we are not the first person to do this. There are a lot of people are doing this right now, but I'm not sure that I have success for drug discovery so far. But I think none of them. But in terms of pharmaceutical research, yes, them are doing right now. You can Google chlorQ and NHP. from the Google. Yeah,
Yeah.
I don't know anymore. So I'm sorry, I can't answer this question
Oh,
in more
okay.
detail.
Yeah, it's all right. Yeah, maybe the question is very specific. Yeah. So we go to the next question. Are the proteins from the two different gene clusters spatially separated? Are secondary metabolism from non-collinear genes less efficient than that from their comparable collinear genes?
So this is another a good question, and we have never talked about that. So it's not partially separated. So if you look at my paper, the publication just we actually have drawn the genome, the genome is circle shape. It's the size of the genome is about 5 megabase pair. And we have in our supporting data, we have shown how far of these two gene clusters separated. It's over 1 million base pair. So it's really far away. So it's not partially separated. And as for the efficiency of collinearity or non-collinearity, the production of the molecule, the production of the secondary metabolite, whether it's efficient or not, I don't know. I didn't do that, and nobody have do that. As far as I realized that nobody have done that. But I believe the efficiency is same. It's same because the gene cluster might be clustered together, but as the center dogma, where the DNA to mRNA and then to protein is floating inside the cells. So when it's floating inside the cell, it can go anywhere. It can be accessed by the non-collinear genes. Yeah. So I believe it doesn't affect much about the efficiency to produce arginine in this case. Non-linear genes. Yeah.
Okay, thanks for your views on this. So let's go to the one more questions on the Q&A box. You have shown that prenyl transferase are precursor specific DMAPP or GPP. I wonder whether prenyl transferase is organism specific. Can Cyanobacteria's prenyl transferase works in heterologous hosts such as plant?
So this question is really new to me. So I don't know what's your idea to put inside the plant. As you know, plant is really a slow growing organism. Are you going to wait a month to see the production of this just for one experiment for a month? So we do inside the E. coli. One reason is we can do it in a short time, like just tomorrow. We do together. We do tonight. After 16 hours or 18 hours, we can check the LCMS. So we can do tonight. Tomorrow we can get the result. So there's no reason why we have to put inside the plant. And I'm sure nobody want to do that because nobody want to wait that long just to see a heterologous experiment. Of course, and I don't think it can work in plants. As you know, plant is what do we call that? Is it there's a prokaryote and eukaryote organism.
Yes.
So it's completely a different thing. The proteins inside the cell, the system model is quite different. So cyanobacteria and bacteria are almost same type of organism, unicellular. So their system is quite similar. So that's why that's how it works, and that's how people design E. coli as a good heterologous model, heterologous host for the expression of the gene. So
yeah,
I think it will not work. It will not.
Okay. Okay, thanks for your idea. So we have many questions from the Q&A box, but let's just go to the participants. Is there any participant interested to ask the question directly to Dr. Phan? You can you can press the raise hand button. Our technical team will promote you to unmute your mic.
Yeah, I saw my supervisor.
Yeah, yes. Can you hear me?
Yes,
Yeah,
we can
we
hear
can hear
you.
All right.
Professor
Thank
Charles.
you,
Yeah.
Pang. It was really nice to hear you talk and present. I see that you have done fantastic work. It's really an honor to see you do so good science. Now, my question is, Pang, at one point you showed, I think it's an enzyme that is a novel enzyme that you have identified, and then that it enhances the biosynthesis of not 5 carbon, but more than that, I think, huh?
Yes.
Yes. Okay. Now, but your issue is you can't get sufficient amount of final product,
Yes.
right? Now,
Yes.
my question is, if you have identified the gene, that particular gene that's responsible for the production of that particular enzyme, I'm sure there are some ways of probing or exciting that gene to express more of that particular enzyme, either by inserting it in an E. coli and using a bioreactor, or some other ways of probing it, with some chemicals and stressing it or something, so that that particular gene could be overexpressed. Now,
Yes.
you could do that if you look at the chemical composition of that particular gene, what sort of amino acid it consists of, and the 3D structure of that. Have you thought about that, and what are your ideas pertaining to this?
Yes. Professor Charles is correct about this. We can overexpress this in E. coli by adding a overexpressed promoter in front of our gene cluster. We can do that, and we will get enough amount of this minor product and do the MR structure or bioassay in future. We talk about that, right? So that's why right now we try to engineer the protein to make it more efficient to accept the another brainy donor, which is GBP. Just now, Professor Charles have mentioned the GBP, another brainy donor. More longer chance, 10 carbon. So if we do that, we success, and we can show people we can engineer the protein to accept a new brainy donor in a more efficient way, and we can characterize that product using MR. So we want to do that in a separate word. Yeah, so we are doing that. So that's why we didn't include this in our publication.
Oh, that means you are looking at biosynthesizing the protein, the enzyme protein itself, because you've already identified that particular protein, so there's no need to overexpress from the organism itself, huh?
Yes.
I think that's one way of doing it. Very, very ingenious way of doing it. Thank you very much. I wish you all the best.
Yes, thank
Certainly
you.
nice to hear from
Professor
you.
Charles. Yeah.
Thank you, Prof.
Thank you.
Charles.
Thank you.
Yeah,
Thank
for
you.
the question.
Okay.
Yeah.
All right.
Any participant would like to interact with Dr. Phan? You can raise your hand. We will take one last question. Yeah, we have this Kwan Hong Ong.
Yes, hello. Hi.
Yes, we can hear you.
Yes,
You can direct your question now.
I have a question that nowadays we are talking about translation research. I know what's the significance of your study in terms of application, apart from publication, and to the environments or community?
I'm sorry.
Can you repeat your question again?
Actually, the last two of my question is in the Q&A.
Oh, really?
Yeah. Just
Wait,
skipped it. Yeah.
I saw. Now there we are talking about translation. Is that one?
Yes. And another one above. Yeah. Can I know how do you discover the gene correspond to your newly discovered compound?
Oh, I didn't see the other question. Can you repeat?
I mean, like, can I know, like, how do you discover the genes correspond to your newly discovered compounds? Like, how do you find that genes? Since like,
Or
it's
how do we
newly
locate?
Yeah,
How
yeah,
do we
locate.
locate?
Yeah.
So how we locate the gene cluster is just now I didn't mention specifically because there's a lot of detail. And of course, you're welcome to see my paper. Just of course,
Yeah, sure.
is a really good paper. So it's everything is transparent. It's mentioned. It's over 100 pages of the supporting data. You can check that.
Yeah,
Of course, I can answer you right now in a brief.
Yeah,
brief
can I
mode.
maybe
Yeah. So
maybe can I have like your email or something so that I can like I can get more information from you
yeah,
from that. Yeah.
sure. You can Google my name, or
Sure.
I can drop you something. Your name, Kui Hun On. Okay.
Yeah.
So to answer your question, to how to locate the gene cluster, first we do the genome sequence, a complete genome sequence, and put inside the in the software Biosynthetix call, and then we do a T plus N using the quality of our core peptide, which just now I show you, RVIPPP, something like that. So by doing that, we can quickly locate the gene. Locate the core peptide from the genome sequence, and that allow us to locate the gene cluster. And after we locate the gene cluster, we do a blast against the NCBI database, and we found there's a high similarity of the HCCF to the planning transferase, like over 40, not really high, like 40%, which is enough to propose it's a planning transfer activity, but in a new way, in a new protein. Yeah.
Oh, okay.
That's how we do that. Of course, you need to use a software. So another question you asking is the significance of our study. So yeah, just now I show you in my conclusion. I'm not sure. Maybe I didn't explain it nicely.
Yes.
All right, I explain again.
Sure.
So we really try to make a biocatalyst, engineerable protein to do that, to let the protein do the work and make the compound, make the molecule, so make the antibiotic. So of course, this is not the only way. In conventional way, people can do that. For example, the synthetic chemists, people synthesize the natural product using a lot of synthetic step, 40 step, 50 step to synthesize one molecule in 1 week. Yeah. So it takes a lot of time, and it uses a lot of harmful solvent, which is organic solvent that is not good for the environment. So we are doing it in a more sustainable way and more environmental friendly way. So that is one that is good for environment. So your question also, what the significance of this? So this protein can be used for pharmaceutical industry. So as I show you, it's very simple. You just have to have the substrate and mix with the protein inside a flask, and for a few hours, and you get the product. It's really fast, and you can get a bunch of this analog, and you can screening for the antibiotic. So this what we do is we propose a platform for pharmaceutical industry for drug discovery process. So I mean, if that success, it will be a pretty big deal, because it will be one of the example for drug discovery process that this platform. So I think it's significant.
Yeah. Thank you. I mean, yeah. Thank you for that's all I think.
Okay. Yeah. Thank you, Kwan Hong for these questions. So I think we have reached our end of the sessions. Yeah, I understand that many participants are very interested on Dr. Phan's research, and he has give very detailed explanations on the cyanobacteria, very good interactions between the participants and the speakers. Yeah, we really thanks Dr. Phan Ching Soon for these sharings.
So it's finished. So I want to add something. So I really want to advertise JSPS. I want to advertise again. I mean, just like I already advertised.
Yes.
JSPS, a postdoctoral fellowship. So please apply JSPS postdoctoral fellowship if you are finished a PhD, you are fresh graduate of PhD. So it's really a good opportunity for, you know, it's like a turning point to boost your scientific career. So with JSPS postdoc fellowship, you're able to work in a top university in Japan. The facility is really good, and you are surrounded by a lot of genius and talented students and also professor. So when you mix with this type of people, and you will become, you will improve. Yeah, I'm not saying you will become a genius, but you will become think like a genius. So this is very good to tackle the research problem and to make a good research paper and to give benefit for the society. And I want to advertise Professor Ogino from Hokkaido University. He's really a good professor. He's actually under the Bill Garvey, one of the pioneer natural product in Cyanobacteria. That's why I mentioned that. So go for him. Yeah, he's good. And also Professor Toshiyuki Wakimoto from Pharmacy of Hokkaido University. He's also one of the genius, and he's really good in biosynthesis. So if you are really interested in genomic way of study of natural product, so contact him. Yeah, that's all.
Yeah, thank you, Dr. Phan. Yeah, I just observed this one last question from the Q&A box. Yeah.
All right, sure.
Very quick. Dr. Phan, have you validated involved MOE-N5 in any of your studies previously?
I don't know what is MOE-N5. Who it is? Can you speak right now?
Yeah, can you raise your hand and ask a question?
It's okay. You can ask. Okay. You can contact me by email if you want to. If you're still interested to ask me this question. You know.
All right. Yeah, the participant also can leave your email in the chat box. So with our secretary will contact you later on. Thank you.
All right. Thank you very much.
Yeah. So thanks, Dr. Phan, for sharing your session with us. So the organizing committee would like to present a certificate of appreciations to Dr. Phan Ching Soon for a very fruitful presentations to community as well as the community around Malaysia. Thank you, Dr. Phan.
Yeah, sure. I can come anytime in future if you want to invite me again. It's my pleasure. It's my honor. Yeah, it's really a good experience for me. Thank you for invitation.
Oh, okay. So, dear participant, we will post the feedback form in the chat box as well as this screen. You can screen the QR code. Please fill in the feedback form for our improvement. The committee will email the e-certificate to you later. The feedback form will close at 1:30 p.m. today. So no certificate will be issued if there is the entry is not captured in the feedback form. Right. And on behalf of I would like to take this opportunity to thank the organizing committee to make this workshop success. I'm sure this event has brought fruitful knowledge sharing to our participants, and we wish Dr. Phan good luck in his future career. Thank you.
Oh, finish.
Yes.
See you.
Thank you, Dr. Phan.
I'm leaving.
Yes.
Okay. Thank you very much. See you.
See you. Bye bye.